Okay, this is a voice recording for your activity known as the Homologene activity. So this will be your first go at making some phylogenetic trees. So there's two main websites that we're going to use. Both of them are free. Both of them should be able to be used on your Chromebooks. Optimally, they will work better on a much more advanced computer, but if the Chromebook's all that you're working on today, then it should be okay. So let's get you oriented to. This is the National Center of Biotechnology Information's website. It's chock full of information, and it's where people like Dr. Hip dump in a lot of the sequences that he's done for all the different species that he's looked at. Chicago Botanical Gardens will do that, Northwestern, all the major institutions, not only in our state, but across the world. So let's start by this tab up here. So this tab right up here is what you're going to hit and drop down. It brings you all these different options. And all we're going to focus on today is the homologene one. So you're going to hit this one, and homolo, if you understand the prefix, means same. And then we're looking at gene, so the same genes. We're going to look at gene sequences that are the same or how close they are the same and generate a phylogenetic tree or evolutionarily related tree for a bunch of different organisms on the tree of life. So let's do the first gene as your packet says, which is GAPDH, G-A-P-D-H, glycerol aldehyde phosphate dehydrogenase. What a mouthful. You're going to do some research. That's part of the components in step 2 of your packet, but let's just walk you through some of the logistics stuff. So I hit the search button, and we pull up, and you can see I've already done this ahead of time before I made the video. Right here, we're going to look at this glycerol aldehyde 3-phosphate dehydrogenase. If we hit that, it takes us to all the organisms they have sequenced for the GAPDH. So you can see that there's color codings. You can look into that. Most of this is based on the protein. So remember, that goes gene turned into a protein, or DNA into protein, or genotype into phenotype. So we're looking at the proteins that are actually made and how similar they are between all these different organisms. Now, if you look in the list, we have H. sapiens, that's you, Homo sapiens. We have P. troglodytes. We have a bunch of other ones. And if you notice, some of them are the same name. That's going to pose a problem, and I'll talk through that once we get there. But it's not too hard to reconcile. So first thing is you're going to have to do is you're going to have to actually download this sequence information. So you just come over here to this download button, click the download, and you come to this main screen. Now, you're not done yet. This is just showing you that, hey, these are the ones that you truly want to download. And the answer is yes, we definitely want to do that. So we're going to come over here, we're going to hit the download. And when we do that, you notice that I'm downloading. It's going to show up a little bit different on your Chromebook. Regardless, it's going to be a text file, which is a very basic file format for computers. It's usually read by a lot of different types of programs, whether it's an Adobe program or a Microsoft program. So this notepad file is going to be the one that we're going to use. Now you have two options. One, you can save this and then load it into your next website, or you can copy this, paste this onto a document, and then just copy and paste it later. So whatever you do, you're going to have to still modify this. And in the directions, one of the things that you're going to have to modify is the name that comes before all of these letters. These letters here are the actual amino acids that are coding for GAPDH. Now, GAPDH is an enzyme, hence the hydrogenase component. We're going to look at how similar they are. So you want to make this so that the tree looks clean. You're going to save this little caret here, and you're going to rename this section for whatever Homo sapien truly is. So you might rename this as human, right? And you're going to go through and do that for all the different organisms. So you're going to have to do a little bit of research and look up this. I don't care if you work with other people, that's fine. But everybody's going to have to do this tree analysis. So everybody's turning in their own trees, and everybody's answering the questions on their own. But you can work together in figuring out what these genus species turn into for a colloquial language, what you would call them in layman's terms. All right, so we got that first part done. Moving on to the second part. If you just type into Google phylogenetic tree maker France, it's the same one that would pull up if you typed in just the HTTP code that I have listed on your actual document. It looks like this, and here it is. So this is done by French scientists. It is an algorithm that pulls out all the possible relatednesses, and using that algorithm, it pumps out the best possible tree using a concept called parsimony, which is the most logical thing is probably the most correct thing. So once we get here, this website is just absolutely fantastic. You come down to this one click. You click on that one click, and it brings you to this page. Now this is where I said either you save the file or you cut and paste. So if I do the cut and paste, I can literally cut my document here, all this code, and I can just copy it, so Control C, and bring it over here to my website, and Control V, paste it right in. Now when I do that, it's all set, and we're ready to go. You could also do the choosing of file, and you could do it that way. If you want to name it Homologene, you can do that. You don't have to, but then I do suggest that you type in your email so that you can go through and get this email sent to you when it's all done. All right, because sometimes depending on how many trees are trying to be generated at one time from this website, it could take a little bit of time. So once you get to this point and you hit submit, you're all set to go. So I'm not going to do that. I'm just going to show you. So it starts rendering, and it's going to work through some curation, and it's going to work through what's called muscling. So muscling is when it aligns gaps in the code so that the codes line up properly, and that's what you can kind of see with this little running, I guess, GIF it would be right here. And it'll keep going into the point where it gets to a tree rendering. Now, I'm not going to sit here as it does that. You're just going to have to trust me that eventually it'll pump out a tree. If you do an email, it will send it to you when it's done, and you can look at that tree, and you're going to use that tree as part of your analysis in class for next week, but also it's going to be that lab homework component that you're turning in as well. So print that tree when you're done, follow the directions, because you're going to have to go back to this website and you're going to have to do a homologene one, but you're going to look at a different gene as opposed to GAPDH, do a little bit of research about those genes, and then finish up your mini little project. Hopefully that makes sense. If it doesn't, just send me an email or post it on community, and we can all work together to solve your problems for the day. I hope the day goes great for you. Look forward to talking about your files and your trees once we come back together on the following Monday. Have a great weekend, and it's been a great Science Friday.